All right, so we've been talking in lecture of the idea that microbes have some options on how they look for food. They can sit and wait, they can move around. They can do some more predatory style hunting, so to speak. Gas vesicles, if you're aquatic, this is a good strategy to get to the food. You can just float usually upward, if you're photosynthetic, of course. But sometimes you want to float downward because as things float down, particles float down and land on the bottom of the stream. That may actually be a really good area. If you don't do photosynthesis, there's plenty of carbon down in that area. And remember that microbes are also hunted for food. They are prey for other organisms. So they have to try to deal with that as well. Yes.
We know that there is like fungi that we're looking at, is that still acceptable for?
Absolutely. Love fungi. So yes, in fact, and with fall, I've been seeing some really fantastic fruiting bodies out in the woods. So discussing them, could they be associated with the tree in terms of a mycorrhizal relationship? And so these types of things. And as you're looking at your site more, if you have questions about your site and want to chat with me, absolutely. I would be happy to give you feedback on some of the things that you're seeing, and is that something you should include or not? So make sure to ask me, and Duncan's a good resource as well. So microbes are capable of assessing their environment very well, and including, of course, nutrients, because they have to gain access to nutrients on a daily basis. And simple experiments in the lab show that they can hone in on, here you're seeing a concentration gradient where you have, they put the common experiment for this is to put some live microbes on a microscope slide in water so that they're free to move around. And then you drop a concentration of maybe a sugar solution, so the attractant here on the edge of the slide, and it starts to wick under the slide in a concentration gradient. Of course, it's diffusing out. And what they're trying to show in this image in figure A is that you have initially the microbes are just kind of seemingly randomly moving around, searching. And then as they hone in on this concentration gradient, they are able to do a more directed movement toward that attractant. Let's say it's a carbon source that they're wanting to use for their nutrition. And so they actually use these words, run and tumble. That's actually when you think about when they have flagella in particular, especially if they have many flagella. Some bacteria have lots of flagella, which is mind-boggling when you look at the complexity of flagella. But they will build many, many flagella, and then they'll all kind of come together physically and start rotating in unison. And that's when you have a run. So that's what they're trying to show here, where the flagella are more with each other and you're doing a directed movement. So that's the run stage. And then the tumble, what they can do is let their flagella kind of move apart from each other, and that causes them to slow down and they can reorient, and then they can start up again. And so we won't get too much into flagella, but flagella are pretty fancy, and they're able to definitely help quick movement, relatively quick movement compared to other movement styles that microbes do. So that's a good way. Another thing to think about is that some microbes have this ability, most microbes have the ability to form biofilms. And so capsules and biofilms are the idea that microbes can synthesize this substance that they excrete out of themselves, and then it coats their cell. And so then we start saying the capsule. Oh, look at the capsule on that bacteria. So here in figure A, what you're seeing, all the black dots are the actual microbial cells, and all that hazy gray around it, that's the capsule. So it's mostly a sugary goo. It's a polysaccharide that they're excreting out, and it's very watery but gooey. And it allows them to, it coats their whole cells, and you can see it can be quite substantial, many, many times bigger than the cell itself. Figure B is an electron microscope picture where you can see the capsule, the light gray around the cells. And then here, figure C, you can see a pretty whopping big capsule. All right, so when you look at individual cells under the microscope, we would say the word capsule. Oh, look at the capsule on that microbe. Cool, look how big it is. And then when you start thinking about, like right here, you see many, many cells kind of clustered together, and you start to see how their capsules kind of merge with each other or coalesce with each other, and it gets even bigger. Then we start using the word biofilm. So when you say biofilm, most microbiologists are thinking about a community of microbes all doing this, and you have this big gooey substance around all of them.
Is this the stuff that you can feel on like cyanobacteria that like sleep breathes like?
That is similar. It's a little different chemical composition, and we tend to not call those biofilms, but it's similar, and it's a similar idea of what it's doing for the micro or for the cyanobacteria, and algae too can do this. So when you probably the most common experience you might have with that is every morning when you wake up and you run your tongue over your teeth and it's kind of, goopy and furry. That's a biofilm on your teeth. The microbes all night long, because you haven't been eating or drinking and washing the microbes off your teeth all night long, they've kind of had a party in your mouth, and they've been reproducing themselves and forming that biofilm. And then when you wake up, you can feel that. And so that's probably one of the most common examples. So depending on where you're reading about biofilms and capsules, they call it different stuff, unfortunately. So some people call it an extracellular polysaccharide matrix. Biofilm is usually what I go with because it's shorter and easier to talk about. And so, but just be aware that you'll see some different terms in your readings, for example. So, but basically what I want you to know is it's a polysaccharide goo. It's a matrix, yes. And they make it themselves and excrete it. And it can be very useful, especially when you start thinking about communities of microbes all doing this in their natural habitats. So here you see this idea of, here they're showing, so maybe think about a rock in a stream, for example, and you have microbes hanging out in the water, and then they will find the rock and they'll start to attach to it. And then more and more microbes come and attach in the same area, and they start forming a population, and they all are excreting that capsule material. And then as more and more of them are there, it becomes a biofilm. And they've done some really cool experiments lately in the lab where they can show that the biofilm really is a three-dimensional structure, almost like I tend to think about downtown Chicago when I think about biofilms, in terms of the idea that there's very tall areas and smaller areas, and like the streets of Chicago. I think about how that is some of the holes here that allow for water flow to come through, nutrients to come through. And so it's not just the solid goo, it's got this internal architecture that allows for water flow through it, nutrients coming through, waste going out. And so it's a very dynamic area, and it's, of course, strength in numbers, having a bunch of you around, all doing your thing, excreting enzymes, you're working as a team to get access to nutrients. And so it can be very beneficial to live in a biofilm. Yes.
Sounds stromatolites in into existence.
Pardon?
Is this how stromatolites?
Yeah, stromatolites are very similar, like with that, they don't really call it a biofilm, but it's an extracellular matrix that they exude, and then it forms this kind of mat of microbes and cyanobacteria, and then they just keep layering on top of each other over the years. So, do they show? Well, let me go to here. So again, it doesn't just happen in aquatic settings, but very often it does. So I'm sure some of you have been hiking in a stream and the rocks are all slippery, and you look and there's like, you don't see any plant life on there. It just looks like a rock. But that slipperiness is probably the biofilm that you're stepping on and experiencing. So here you're looking at iron oxidizing microbes. So they actually strip electrons off of iron, and it can cause this rust color in the water. Sometimes it can be quite substantial depending on those sites. One other thing about biofilms, I just want to point out, they don't really show it here directly. They're showing the buildup of how you get a biofilm, But we now recognize that this is not permanent. So in natural settings, you can imagine a situation where maybe this habitat starts to become low in nutrients. And then it wouldn't make sense to stay there. And so they can disperse. It's not permanent. So they can leave this population, leave the biofilm, and hopefully find a better habitat and do it all over again. But most microbiologists think now that the biofilm lifestyle, at least for some of their part of their lives, most microbes in natural habitats do this. It's not just a special little group that can do it. And so this is a standard thing. It's a problem from a human perspective in some ways. For example, if you've taken an antibiotic and one of the pathogens in you is forming that lovely thick capsule, you can imagine how the antibiotic might have a tough time diffusing through that capsule. And that's true. So I once got a bunch of microbes from the community, Houghton Community Health Department. And oh my goodness, I'm streaking them out. I'm like, wow, almost all of them had capsule production. And they were isolated from patients that, it was a pathogenic version of that species. And so if you isolate something like that from a patient in the hospital setting, it's like, uh-oh, that's a good strategy if you're a pathogen, not so good for the human because it makes it harder to treat it. It also, can keep them protected against other chemicals. It can help them avoid drying out. It keeps them moist. And so they don't dry out as quick if they have all of a sudden a drought period. So this capsule production can be very useful from their perspective, but it can cause problems. They're also a big problem in like pipe systems in your home, in the city pipes, because biofilms will just start to, layer down on the inside of the pipe. So city workers and, people that have to think about water flow through the city, this is something they have to think about and deal with. So good and bad.
So you're talking about it being, is there something that they can give those patients to like attack the
Yes.
capsule itself?
Yes, they're starting to really, there's been some really good work. I think a paper that was published a few years ago where they're looking at disrupting the biofilm and they're having some good success with that. So it's not, they're working on it and they're having good success. So it's not like you're doomed if there's treatments available. So. Okay, so what else do we have going on here? So you can imagine as you're a tiny, tiny little microbe living in your natural habitat that it's not always roses and that you're going to be dealing with situations sometimes, many times through the day where you are faced with a tough time. Maybe it's a drought, maybe it's lack of nutrients, maybe it's too much waste material around you and you're starting to have a toxic buildup of your own waste essentially. And so microbes have to try to deal with that. This is probably the most extreme way to deal with it. If they encounter a tough time, some microbes can form endospores. And endospores are, as far as we know, the most resistant biological thing that we know of. It is amazing at how resistant it is to all kinds of things you can throw at it. Heat, pressure, nasty chemicals, drought, UV light, and it laughs at you. And so it is amazing. And it is a process for the microbe to form it. It's really only, I don't have it on here, I don't think. Well, maybe it's on another slide, I'm just forgetting, but.
You see endospores, does that mean like inside?
Yes, whoops. So gram-positive, that's supposed to be a capital G with a plus sign. Gram-positive bacteria are the only ones that we know that do this. You're going to learn about gram-positive versus gram-negative on Friday in the lab because we're going to work through the gram stain process, which is a classic micro lab technique that you need to get comfortable with because you'll probably use it through the semester. As far as we know, only gram-positives do this, and it's inside, endo, within. And so some spores, a lot of other spores they're not done this way, the way the endospores are produced. And I'll show you a picture. So basically it's all done within the vegetative cell. And so the, the living vegetative cell is like living its life, and then all of a sudden it's like, oh boy, I don't have any water around me. And I still don't have any water around me, and I still don't have any water around me. After a while, they're gonna make the choice, so to speak. I better kick into endospore production, and it's all formed within the cell. It's done inside. It is expensive to do though, so that's why I'm trying to say that it takes them a while to kick into this because it's energetically expensive. It takes a lot of resources. And once they start the process, they can't back out of it. So let's say they're experiencing this long drought and they start making endospores, and then whoosh, nice rainfall event. They can't go, oh, I'll just quit. It doesn't seem to happen like that. They have to take it to completion. And so once they start it, it's a cost to them, and hopefully with a good payoff at the end. But lots of genes get turned on, very unique proteins and chemicals get made in order to, basically what they're doing is forming a version of themselves inside layers and layers of very complex and tough substances. And then they start to dry out. They'll dehydrate themselves. And then when the vegetative cell, once it's all set to go, the vegetative cell will break open and the endospores will be released. But essentially that endospore has a vegetative cell inside of it. It's just way dehydrated and essentially gone dormant. And so it's really an amazing process. And so non-reversible, we've talked about that. It can't go back once they start it. So it's really, I won't go into all the details of the process, but it's a big deal.
You mentioned that, what are they able to do while they're doing this? So are they able to still gather food? and
The
whatnot?
vegetative cell?
Yes,
No, they're pretty much focused.
okay, so it's just that.
Yeah, they're
They're
pretty
really-
much like, okay, endospore. They're pretty distracted, so to speak, by, I gotta form this endospore. Because times are tough. I need to do this in order to survive. So it's a survival strategy. This isn't a way to reproduce. This isn't reproduction. They're just forming a version of themselves that can get severely dehydrated within layers and layers of very complex proteins that allow it, and other sugars and things. And so it gets them into a state that allows them to deal with, maybe months of drought, and they'll just be fine. And so here, just a little bit, some of the unique chemicals that they find. So for example, this one, dipicolinic acid, is formed in order to stabilize the DNA. So if you're going to severely dehydrate yourself, I mean, DNA, proteins, enzymes, they all have to be protected, especially the DNA. I'm not getting into details of this, but this is just a visual showing, they actually have mapped it out into the different stages, what's happening in every stage. You don't need to know this. I'm just trying to show you a visual of that. This is all internal. They're trying to show that there's layers upon layers forming that, they call them the coats. And then there's the so-called core region, which is basically a vegetative cell that's highly modified and dehydrated so that it can withstand being dormant. Okay. Under the microscope, when you're looking at some of your environmental samples, if you take sample and look at it under the microscope and do like a simple stain or gram stain, you may see something like this, where it has a non-stained region that kind of bounces the light and it's pretty bright. That's a dead to give away that it's an endospore. Endospores don't like to take up stain unless you really force the situation. So just an ordinary staining procedure, it won't stain and it'll show up like that. So that's a dead giveaway. Here, this is nice because you can see this cell kind of in the middle of the image where the spore is still inside the cell. And then you can see on some of the other areas of this slide that the spore is free. So that one's looks like it's free and the vegetative cell has already gone away. This one's still in the vegetative cell. Under the electron microscope, a spore looks like this, where you have multiple layers. So many, many layers, making it very tough. And so definitely it can be advantageous if you have a habitat that has gotten very adverse for you. And if it's gonna be a while, that it's gonna stay that way, then it makes sense to do an endospore, because endospores are so capable of resisting all kinds of things and being fine until things are better. Maybe a rain event or a big flush of nutrients, and then that spore can rehydrate, get active, and it grows out to be its normal size, and then it starts living its life. All right. And so if you have a drastic environment that's going to be that way for a while, it makes sense to do endospores. But if you have environments that maybe there's a little bit of a drought, but then there's a rain, and so it's a tough time for a little bit, but not a long stretch, endospores might not be the way to go. There might be some other methods that you may want to try before you kick into endospore production. Also, if you're in a population and you have a tough time, like say there's a little bit of a drought, you may have neighbors that aren't quite so good at surviving that. So as they're dying, they break open. So then that's a source of nutrition for you. So there's that dynamic going on that, you're going to have some neighbors dying off that then release their nutrients, and you can take advantage of that. So there's endospores are not always the way to go. And like, here's an example of some other spores. So the streptomyces are the group of bacteria that I do a lot of research with. And you have experience with them too, whether you know it or not. If you've taken antibiotics, I would wager that you've taken an antibiotic that's been produced by streptomyces. They still grow them up and make the antibiotic, and then they purify it and put it in pill form and give it to you. So they're an interesting microbe because they have a very complex life cycle compared to like E. coli. When E. coli wants to reproduce itself, it just gets a little bigger and splits in two. This one has this whole process where it has a spore that will then land somewhere suitable. It grows hyphae, which is unusual for a bacteria. And then if times get tough, it'll start kicking into the hyphae, start to segment. The little crosswells form down the length of the hyphae, and you get the spore formation. And then those will just break free and hopefully get dispersed somewhere. Those are not endospores, all right? It's not the same thing. And they're somewhat resistant. They're pretty stable, but not like an endospore. And so I love working with them because they do form spores. That makes them easier to work with in the lab. It also makes them more attractive for like real life application, because you can give a farmer a bottle of spores and say, sprinkle this into your field and you're good to go. You don't have to worry about giving them living microbes with a short shelf life and dealing with all of that. And so these bacteria are very interesting from many different perspectives and a lot of applied uses. They can be capable of that as well. So sporulation, there's different kinds of spores. Endospores are the best in terms of just how tough they are. But there's other strategies. So there's been some interesting looks into this. How do they deal with different starvation? So in the lab, starving microbes is pretty easy to do. You just don't feed them very much and they start to respond to that. One of the studies was showing that some bacteria can actually release a protein that they call killing factor protein. And what it does is it attacks nearby cells so that it causes those cells to lyse. And then the microbe can take up the nutrients. And so this is, a little bit darker way of making sure that you stay alive. It's actively killing off your neighbor, so to speak, so that you can get access to their nutrients. Some people in the literature actually use the word cannibalism, which is an interesting thing, but I thought that was kind of humorous that they were equating it with that term. But so these proteins, bacteriocins, have been really well studied. This is a group of proteins that many bacteria can make. And so the Gonzalez was an early study, and then they started calling them bacteriocins because they realized that it's kind of a common strategy out there. Many bacteria can do this. but it's targeted. So these proteins don't just kill anything. It's very targeted, and it seems to target closely related strains to the producing organism. So if you're Pseudomonas aeruginosa and there's other Pseudomonas around you, especially other Pseudomonas aeruginosa strains around you, you can use this protein toxin to effectively lyse them open, gain access to their nutrients. So there are those types of interactions going on when they're dealing with tough times. They can also, what we say, go into a resting state. So this is not endospore production. It's just their vegetative cell starts to find ways to slow down and really, I guess, I equate it with kind of going to sleep, but it's not an endospore idea. So they actually can change their cell shape or size, I should say. They can get smaller under starvation conditions. They slow down, so their growth rate is greatly slowed down, and sometimes they just won't bother doing any kind of major growth at all, and they won't reproduce. Their physiology slows way down. They start to tap into anything stored inside of themselves. So, we store glycogen and fat for our energy. Microbes, believe it or not, they have some ability to store up some nutrients. They're tiny, but they can pack in. It's pretty common that they'll have complex carbs in their cell, and they'll have phosphate molecules that they've kind of pieced together into strings of phosphate that they can then cleave off and tap into. And so there's other examples too. Those are just a couple. So they have these internal storage molecules that they'll save for a rainy day. And then once things get tough, they'll start tapping into it if needed. They start to really be carefully deal with their protein turnover. I'm not going to get into that, but it's basically slowing things down, really trying to keep things. They don't want to really go through protein synthesis if they don't have to. And so it really gets tightly regulated. And then they try to have a situation where they're poised and ready to take advantage of when good times come about again. So they want to quickly capitalize on any inrush of nutrients. So they have to have those types of proteins and enzymes kind of ready to go so that they can immediately start becoming active. Many bacteria that have the ability to morph their shape, they might form, this is a fancy word, prostike, fancy word for stalk. They'll form a stalk. And so some of them will shift into that shape so that they can attach. And so if I'm thinking this is usually they're studying aquatic microbes where they may come across an area of the stream, for example, that's like, oh, wow, this is really flush with nutrients. And then they can attach there and forage and feed, so to speak. And then if they have to, they can detach and go on their way. And so different strategies for sure, depending on what species you're looking at. But if you think about it, it makes sense that they have several strategies in play to deal with tough times. So spores definitely are an option for many microbes, but not all. Some bacteria can't form spores of any type. And so they're stuck with some of these ideas. Some microbes really change their shape in an interesting way, and then they really pack on flagella. It's amazing. This is an investment, so it's got to be kind of tricky for them, where they have to time this just right. But we see microbes that the most well-studied genus is Proteus. This is Proteus mirabilis. That's probably one of the heavier studied swimming and swarming type of microbe. So basically what this is trying to show you that depending on what state they're in, they're going to physically look different and act different. On the left, you see the swimmer cell and what they're, well, I'll get into a different picture, but basically they're showing it's shorter, modest amount of flagella, they're clearly capable of moving around. And then they're showing they say environmental signal. So in the lab, when you're studying this, you stress them usually with carbon restriction. You starve them from carbon. And then they start to elongate. They make way more flagella. And then they're starting that in that state, they're called a swarmer. And then you can see here down at the bottom of this column, it says swarming. Chemotaxis just means they are capable of sensing chemical gradients around them and going toward the good stuff. And then it says coordinated cell-to-cell communication. So when they're in a swarmer state, they tend to hang out in groups. And it's kind of like, I use in my head the analogy of a wolf pack, where they start to pack up in groups and hunt. And that's what it, when I've seen video of this, that's what I think of. And so when they get into that swarming state, they usually, it seems like they need to have a solid surface as a way to deal with this shift. And so some kind of physical contact that they need in order to start this process of going into the swarming state. And then those cells get much longer, lots more flagella, and then they swarm. They actually swarm over the surface with their flagella. So they're not really swimming. They use their flagella to make contact, and then they swarm across the surface. Of course, in the lab, they use auger plates to do this kind of study. In experiments, they see that if they're just a lone swarming cell, even though they've elongated and they have all those flagella, they don't seem to do anything. They need that pack. They need cell-to-cell communication. They need to coordinate and go, okay, let's go hunt for food. And then they'll start swarming across the surface as a group. So at least with all of the lab-based experiments, they don't see individual swarmers doing any hunting behavior. They just kind of sit there. So that's why they say there needs to be some kind of cell-to-cell communication among them to coordinate and decide that they need to start swarming as a pack. So you can, in the lab, when they do these experiments, they can go back and forth between these states, swimming to swarming, swimming and back and forth. And so I'm not going to ask you the details here about this, but just understand that it can go back and forth. And in the literature, you'll see these words, differentiation and consolidation. I don't care that you know that. Just be aware that they, of course, have to give you some jargon to throw at you, but basically understanding, yep, you can go back and forth, depending on the environmental conditions that they're constantly assessing. So this is trying to show a little bit more about the cyclic pattern of it. This is one version of swimming and swarming across an auger plate. So what they did here was they took a loop and they took a gob of Proteus and they spot inoculated it right in the middle of the plate, and then they incubated it. And what happens is they, start to take in nutrients, reproduce themselves, but then in that patch of agar, eventually they're going to use up all the nutrients, and then they have to move out from that point of inoculation to go find more nutrients. And so they'll start to go into the swarming state, and then they'll swarm, kind of pulse swarm for a little bit. And then you can see this kind of demarcation right there. So then they slow down and kind of stop for a while. They found new food, they forage, and then they go out again. And then, so you just see this kind of bullseye pattern of them moving along, stopping for a bit, moving along. And it's all the way almost to the edge of the auger plate here. And so that's a classic pattern to bacteria that can swim and swarm. So if you think about it, that's a pretty effective strategy to beating tough times. Here, after a while, it's going to get tough for them because they'll have used up all the nutrients. And so to be able to swarm as a pack and coordinate their efforts. Now, this is nutrients trapped in an agar, but you can imagine more in a natural habitat. Maybe there's other bacteria in the habitat where they can use this kind of pack swarming to successfully get to those cells and overwhelm them and kill them off by releasing toxins and other things. Let's see if this video works. I don't know if it will. Let's see. I'm gonna go into oh. Is it just me? But I mean, don't you think about wolf packs for something like that? Kind of analogous, don't you think? So again, kind of demonstrating in a way that a lot of the concepts that you learn in a more traditional ecology class, you can apply it. Many of them, they're finding it works the same at a micro scale with the microbes. So trying to also pull up a video here that I was wanting to show. Whoops, not that one. I'll show that one at some other point. Oh. No. One on twitching. Bacteria actually twitch. Twitching motility, they call it, because of the way it looks under the microscope. But that's not what I'm looking for. Here it is. Okay, let me see if I can. So this is an agar plate. The mutant is on the right where it can no longer swarm. And then they spot inoculated the wild type on the left. So they've sped it up, of course. They have the time scale there. What was that? 18 hours roughly? So those patterns, look for those patterns when if you end up doing any inoculating of samples onto plates, especially from environmental samples, I tend to see that finger-like growth like that. It's many bacteria can do that as they do the swimming and swarming movement across the plate. So that's a fun one to look for when you're looking at your plates. So they initially, when they found this swimming and swarming, they thought maybe it was just Proteus that can do this. And so they thought it was kind of a one-off, unique to Proteus. And then they realized, and you don't have to write all this down, of course, it's in the posted slides, but they realized that, oh, there's a lot of different genera of bacteria that can do it. And this isn't even a complete list. And so the fact that so many unrelated species can do this, leads you to think that it's an important adaptation to their habitats, and it's something that has evolved amongst all these different species based on the selection pressures of their habitat, that it's conferring an advantage to them. And so, definitely when they've studied it in the lab, it's pretty efficient to group up like that and move across surfaces and hunt for your prey and get access to nutrients. And it does at least ensure that some of you survive, to pass on your genetic inheritance to your offspring. And so they are thinking that this is a strategy that many microbes in natural habitats will employ for part of their life. Okay, so some very interesting ideas in terms of ways to deal with tough times, get access to food. Here, this table is looking at different habitats. So you have, a lab rat E. coli, so to speak, and you can get E. coli to reproduce itself. So this column that says doubling time, what that's pointing or thinking about is how long does it take to double its population? Well, that's why it's doubling time, but if you know what you're doing, you can get E. coli to double its population in 20 minutes. That's fast. That's in ideal conditions. So we do that a lot in the lab when we're growing up E. coli for your needs. Didn't you work with E. coli on Friday? You did some streak plates and spread plates. So yeah, we actually set it up the night before and let it incubate overnight. But if we wanted to, we could have done it that quick and gotten you enough microbes to work with. But you can see other habitats much longer. The human intestine, it takes 12 hours to reproduce, especially if you don't eat. Feed your microbes. Make sure you're feeding your microbes through the day. And then you can see some other interesting habitats and the variety of time spans. And then you get into days, the soil. Okay, the soil is a little bit more, can be more hostile, I guess, in terms of you don't have a perfect life and all those sweet things around you to be super efficient. And then look at down here, the deep subsurface of the soil, 100 years to double your population. That's slow. And then look at duration of dormancy. So now they're saying, okay, if you're dormant for a long stretch, how long is too long before you can't revive those cells anymore? And with Clostridium in a test tube, someone grabbed it out of the lab after 34 years and it was still fine. But look at here, Lake Vostok, beneath the Antarctic ice sheet, more than 1.4 times 10 to the 5 years. So that's 1.4 with 5 zeros after it. And look at all the way down, Precambrian salt crystals, 250 times 10 to the 6th years. So they were able to revive those endospores after sitting that long in salt crystals from the Precambrian era. That's phenomenal. Just phenomenal. And so endospores, I tell my Gen Micro students this too. I have a love-hate relationship with them. You gotta respect this. I mean, this is amazing. And yet, if you work in a lab where anyone's working with endospores, you might end up hating them because they can contaminate your experiments really easily. I had that happen when I was a grad student. A fellow grad student was working with bacillus species, which form endospores. And those endospores got into my experiments and contaminated them because we shared some common equipment that didn't get disinfected. Slowed my research down. I lost about half a year because I was troubleshooting all these issues. Finally got it figured out. And the last thing that endospores were contaminating in my research lab, the last thing I found, finally cleared it all up. They were floating around in my beaker of ethanol, ethyl alcohol that you use to sterilize your utensils, your glass spreaders in lab. Remember, you were soaking them in beakers of ethanol before you did the spread plate. the endospores were just floating around, happy as a clam in my ethanol. Just for grins, I took a swab and swirled it around in the ethanol and did a streak plate, like, I'll just see if there's anything in here, and it grew. And I showed it to my PhD advisor and they're like, what? Like, yeah, it was in the ethanol. So no wonder I kept getting contamination. So after that, I didn't share my ethanol bottle anymore. It's like, no, this is mine. No one else can use it. I thought this was interesting. Brewing companies, they used a 45 million-year-old yeast extracted from amber. That'd be a pretty cool claim that you could say about your beer. I thought that's good capitalism right there. So finally aged. So I don't know if it's still in production, but I thought that's a great little hook for anyone sciencey who likes to drink beer. I think that would be a good strategy. All right, we'll stop there. Tomorrow, I do need to talk to you a little bit more about physiology. And then we'll start getting into habitats after that.